Hi, I'm Becca Richman, and I am a senior at St. Paul Academy in Summit School, and I have spent the last 8 months researching how light color impacts the variegation quantity of Epipremnum aureum leaves. So, let's break this question down. First of all, what is variegation? Variegation is defined as the presence of multiple colors on the same leaf. This can be splotches, lines, dots, all of the above. In Epipremnum aureum, it is areas of white and areas of dark green on the leaf, both being present on the same leaf. And variegation can be of genetic or environmental origin. So in some species, it is an adaptation that has come with the environment, and in some species, it is of genetic origin, which means the plant genes code for areas with non-functioning chloroplasts. So chloroplasts that do not absorb any light in the visible light spectrum because there's no chlorophyll A in them, and they simply reflect that light back to the human eye, making them appear white. That's what's going on with Epipremnum aureum. Why is variegation valued? Why is it something that should be studied? So variegation is highly valuable to plant consumers. So in indoor plant communities, it is understood that is the challenging plant to do and like the challenge to take on, and that you're a real plant owner when you can take care of variegated plants. And in general consumption of plants, it is the second most important consideration among plant consumers when they are choosing ornamental non-flowering plants, which means that when plant retailers can sell highly variegated plants, they can make more money. So, what impacts variegation? What do plant retailers need to know to be able to sell these variegated plants? Well, it is understood that light environment, defined as light intensity, which is the amount of photoactive photons in the light environment, and light color, which is the visible spectrum of light, the wavelength of light that is there, impact variegation. It's understood that light intensity impacts variegation, but it's not understood if light color has an impact on variegation. And Epipremnum aureum, pearl and jade pothos, as it's commonly named, is the most popular houseplant in the entire country. And for these reasons, it's really important that we understand what's impacting Epipremnum aureum. How light color, this huge part of the light environment, which is key to not only variegation, but growth, development, and function of all plants, specifically variegated plants, what impact that has on the most popular and most purchased houseplant in the United States. So, I sought to find an answer to that question. And my hypothesis is that variegation will increase under white light. It'll increase the most under white light because it has all of the light that it could possibly need to do photosynthesis. So it's okay if it has non-functioning regions on the leaf where photosynthesis isn't happening, variegated regions, because it doesn't need. Then, based on the chlorophyll A absorption spectrum, I hypothesized that blue and red light, so this peak here and this peak here, that variegation will increase under those as well because there's enough light that is really well absorbed that the leaf will be able to undergo as much photosynthesis as biologically necessary and be able to have variegated regions. Won't need them all to survive. But then when we get into the orange and green light, which is my two other experimental groups, that light isn't absorbed very well. So the leaf is going to need every single chloroplast absorbing light that it can. So under those, variegation will decrease. So how did I go about answering this question? It was a pretty simple procedure where I constructed a light infrastructure in a Conviron A1000 environmental chamber set to 25 degrees Celsius, 70% humidity, and 16-hour moderate light intensity days and 8-hour light intensity 0 nights. So it was basically a greenhouse environment that facilitated plant growth, which they were left in light environments where there was only one color of light. So red, blue, orange, green, or my control, which was white. So they were left in there for 21 days. And with ample water, they were kept at 120 mils of water per leaf, 120 mils of liquid, 100 mils of water, and 20 mils of fertilizer for 3 weeks, 21 days. And on days 0, 7, 14, and 21, the variegation quantity of any existing leaves in that experimental group were quantified through a procedure outlined in Lee et al. 2007, simple and effective method for quantifying leaf variegation. So as you can see here, I took black and white scans of all of the leaves, and then I put them into Photoshop. And I outlined the leaf with the select tool, recorded its area in pixels, then did the same for the variegated regions. And then I created a percentage based on pixels that the leaf was variegated, and I recorded that for every single leaf for all 21 days. So, results. I found that light color has no significant impact on the variegation quantity of Eoram leaves. Plant retailers don't have to worry about it. So let's dig into these results. Let's look at the graph first. So these are the 5 different groups I had. And as you can see, they all decreased in variegation, but the error bars are overlapping, and the p-value for all of these groups was 0.35, which means there's no significant difference between the groups, which means there's no significant impact of light color in general on the variegation quantity of Eoram leaves. You'll see some weirdness in my red data because out of the 10 plants that the red group started with, only 5 survived, despite ample water and resources and oxygen, or carbon dioxide. And then, just to make sure, I did t-tests for each of the white light, which is the control, with each of the colored groups. So blue, orange, red, green, just to see if maybe just blue light or just red light or just orange light had an impact on variegation. And because these t-test values are greater than 0.05, they never did. So we know that light color has no significant impact on the variegation quantity of Eoram leaves. And this makes sense because the variegation is of genetic origin. The light color that a plant is in for 21 days isn't going to change the genetic makeup of a plant. That makes sense. And this does not support my hypotheses at all, but it does answer my research question, so we got an answer. We know that when plant retailers are going to grow plants, they can just focus on the light intensity that the plant is in. They don't need to focus on the light color to be able to provide the best variegated plants possible. It is also important that this study is conducted in larger groups with more variegated plants because this is just one plant and this is just one period of time. And to really have a correct interpretation of how light color impacts variegation quantity, we must do this study on a bigger scale and with many, many more plants. But this is a great start. And then these are my resources that I used in this presentation. Thank you so much for your time. I really enjoyed doing this research. Thank you so much.